go live, I'll just press a button.
Okay. Sorry, everyone. Technical difficulties.
Sorry, I'm not sure. I thought it was working.
Yeah.
Okay. Yeah, it's working.
Ah. Sorry, everyone. Technical difficulties.
Okay, so Bruno, please.
Okay, let's get started. Thank you for letting me chair this in Naotsugu. So I attended a couple of these, or one of these in the past. I gave a talk, and Naotsugu invited me to chair this one because of the subject of general anesthesia, which I work on in fruit flies. So it's a subject of great interest to me that I've worked on since my PhD days, actually, on and off. And I think the principle of these talks is that they should be short, 15 minutes. Maybe 20, primarily just a lot of discussion afterwards. And we'll have first a talk by Katja Valli first, and then followed by Rob Sanders. And they're both on anesthesia in some way. Katja works more on dreaming, so she's at the University of Turku and just got promoted, I heard, at the University of Skövde in Sweden to full professor. And she'll be trying to link anesthesia experiments to dream content, and I think Rob Sanders will do the same, but be talking more about how general anesthetics disconnect consciousness. So that's principally the main things that framing the talks for today, and I think we'll just start and get going so that we can hear what both of our speakers have to say and leave some good room for discussion afterwards. So take it away, Katja.
Thank you, and thank you for inviting me and for the nice introduction. So let's see if I succeed in sharing my slides in a presentation mode, hopefully. And as our chair mentioned, my background is more in sleep and dream research than in anesthesia research, although in the past 10 years, I've had this really wonderful opportunity to work with anesthesiologists who are interested in the mechanisms of consciousness and kind of extend the methods that I've used in sleep research to study subjective experiences during anesthesia and also the neural correlates of these experiences. So I'm trying to link the subjective experiences that we have during physiological natural sleep to experiences that we have under general anesthesia. But I'll start with the definition of consciousness, because in different fields and in different contexts, different definitions for consciousness are used, and that can cause quite a lot of confusion. And in the definition that I use is that consciousness basically means that the organism has some sort of an experience. It feels like something for the organism to be it. The organism has phenomenality, and this means that the organism has consciousness. There are then different forms of consciousness, for example, reflective consciousness or self-awareness, but these build on the phenomenal consciousness. And if we define consciousness this way, it is accessible only to the person who is having the experience, or the organism who's having the experience. And in fact, it turns out that a person's responsiveness is a rather poor correlate of assessing consciousness, as has typically been done, for example, in general anesthesia. If a person doesn't respond, the person is presumed or has been presumed unconscious. However, our consciousness can be either connected to the environment or disconnected from the environment. And when we are in a state of connected consciousness, we are aware of external stimuli, we are behaviorally responsive to it, and we have these internal subjective experiences only accessible to ourselves. In the state of disconnectedness, either we are fully unconscious, meaning that we are not aware of external stimuli, we are not behaviorally responsive, we don't have any types of experiences, but it is also possible to have a condition called disconnected consciousness, where the contents of consciousness are present, but they are not connected to the external environment. So we are not aware of external stimuli, and we are not behaviorally responsive. And then in consciousness research, the problem is, how can we tell whether an unresponsive person is in a disconnected state, having disconnected conscious experiences, or is fully unconscious? And the problem is, how do we distinguish these two conditions? And the only way we can currently do that, because we do not yet have the neural correlate or perfect neural correlate for the presence of conscious experiences, is to try to wake the person up and get a first-person report. But first-person reports are not always reliable, and they are never verifiable, in a sense that because the person is the only organism that has access to the contents of its consciousness, we cannot verify what is going on in that person's mind. And when we try to tap into somebody else's experience, we are dependent on the recall of experience and the self-report of the experience. And for example, if we try to assess experiences during unresponsive states like sleep and anesthesia, so we are interested in these experiences, the question is whether there is an experience to begin with, and if there is an experience, what is it like? However, to gain access to it, we need to wake the person up, and already the state change in itself has an effect what the person is able to recall and report of the experience. In dream research, it's been shown that when we wake up slowly and spontaneously, we tend to recall dreams, more dreams, than if we are woken up quickly and forcibly. The memories of dream experiences are subject to different types of memory biases, and especially with when the time lag from the experience increases to the report, our memory forgets details of the experience, maybe the whole experience. And this becomes especially problematic when we are using anesthetics, because anesthetics have amnesic effects. So they tend to increase forgetfulness and forgetting of the experience. It's also important when we are asking about people's experiences, how we probe it. Who asks the questions? Are they standardized? What types of questions are used? For example, if we ask a person who has been woken up from sleep, tell me everything that was going through your mind, or we ask, were you dreaming? We actually get slightly different responses because of how people understand the questions. So it's important to think about how we try to tap into people's experiences. And in the end, the data that we get is a self-report of the experience, and they are always subject not only to forgetting, filling in the gaps, but also depend on the motivation of the person to report. If there's content that might be embarrassing, they might be willing to censor some of the content. And of course, people have different self-observation skills and descriptive skills to talk about their experience. So basically, the verifiability and reliability of first-person reports poses a problem for all research that is done on consciousness, but especially when we talk about sleep or anesthesia, where the amnesia and forgetting is a big problem. And of course, if a person cannot give a report or says that there definitely wasn't anything going through my mind before you woke me up, is this a reliable report of unconsciousness? So can we ever even verify that a person has been unconscious, or not just, forgotten all the experiences that he might have had? Why then is it important to try to be able to distinguish connectedness from unresponsiveness rather than just kind of scientific curiosity? Is of course that this has clinical significance. Like patients with disorders of consciousness or patients with locked-in syndrome might be misdiagnosed. So these patients might be fully aware of what is going on in their environment, just completely unable to respond. Or there are rare cases of unintended intraoperative awareness. So being awake during anesthesia, having connected consciousness during general anesthesia. And although this is quite rare, this does pose a clinically significant problem. So if the higher estimate would be accurate, this would mean that 1 to 2 persons out of 1,000 would be aware during general anesthesia, which would mean hundreds and thousands of people basically every day. And what do we know about, do people have experiences during anesthesia? Some of these results come from studies where an isolated forearm technique has been used. So basically in isolated forearm technique, before the neuromuscular blockade is introduced to the patient's arm, they get a pneumatic cuff, a pressure cuff, that prevents the neuromuscular agent for reaching the hand, which means that the patients can move their fingers and their hand. And when they are presented questions during anesthesia, for example, do you hear me? Do you understand what I'm saying? Squeeze my hand, squeeze your hand once or twice if you feel this or that. So basically patients can respond during general anesthesia if they hear the questions, if they understand them, and if they are motivated to answer. And there have been many different studies utilizing this isolated forearm technique. And the most recent meta-analysis that I found suggested that even up to 1/3 of the patients where this IFT technique has been used have responded either during anesthesia induction or during anesthesia maintenance. And only some of them have explicit recall of their experiences afterwards. So in anesthesia, it seems that when there is unintended intraoperative awareness, there might actually quite often be amnesia for the event. And in fact, the current anesthesia depth monitors that are in use don't perfectly distinguish these IFT responders from non-responders. So distinguishing connectedness from unresponsiveness is a clinically significant endeavor, and sleep and anesthetics can give us a naturalistic experiment or an experimental tool to reversibly try to modulate consciousness and responsiveness and to investigate the neural correlates or neural mechanisms of consciousness. And Rob is going to talk more about this, while I will focus more on the first-person reports in anesthesia, because we still need the first-person report to assess whether a person has had conscious experiences or not. And as I mentioned, my own background is in sleep and dream research, and recall of dreams, content of dreams has been studied for, systematically for 170 years. And of course, for much longer, people have been interested in dreams during sleep. So we know quite a lot about the recall frequency of dreams from different sleep stages, the content of dreams, and notably both the recall frequency and the richness of our dream experiences vary by sleep stage, and they vary especially by time of night. So when we are awakened from rapid eye movement sleep, we are more likely to recall dreams than if we are woken up from non-rapid eye movement sleep. And also, the longer we sleep, the more likely we are to remember our dreams from both of these sleep stages, and also the longer and richer the dream reports tend to be. The average recall rate from non-REM sleep awakenings is about 40-45%, while from REM sleep awakenings, more than 80% of people remember dreams. So this basically means that our conscious experiences are not abolished even during the deepest slow wave sleep. When it comes to anesthesia, in fact, there are quite a number of studies that have looked at patients that have undergone clinical general anesthesia and an operation. And in the post-anesthesia interviews, 13 to even up to 55% of patients have reported that they did experience dreaming before awakening. And also when patients have been undergoing responsive sedation, so they have actually been on the brink and still being able to respond to questions, they've also reported these dreamlike experiences with relatively similar incidents to non-responsive general anesthesia. But of course, here the problem is that the patients have undergone surgical operations. They've gotten various drugs, so not only a single anesthetic. And of course, they haven't been able to be interviewed immediately after the anesthetic administration has been terminated. So there's always a recovery period after clinical general anesthesia. And this means that the reports that research are getting are subject to the amnesic effects of the drugs and different memory biases. And it has actually been suggested that maybe dreaming takes place during the recovery phase when the anesthetic effects are dissipating and the kind of unresponsiveness is turning into natural sleep. So that maybe dreaming doesn't take place during the administration phase of the anesthesia. And well, we can tap into this with experimental anesthesia. And there have been a few studies, earlier studies, that utilized delayed interviews. So after the study participants had spontaneously recovered, and in fact with contradicting results. In one of the studies that I was involved with, we used dexmedetomidine, propofol, sevoflurane, and xenon to render our subjects unresponsive. And when they woke up, about 60% of them recalled dreamlike experiences. Experiences were most often recalled with dexmedetomidine and least often with propofol. And we also noticed that about 1/3 of these dreamlike reports included references to the research setting, to the people, to the electrodes, to the nurses, and so on. In another study, propofol, xenon, and ketamine was used. This study actually looked at the perturbational complexity index during unresponsiveness. They also interviewed their participants and noticed that none of those participants who received propofol or xenon recalled any experiences, while everyone who got ketamine reported long and vivid dreams. And 4 out of these 6 actually had hallucinations about the research surroundings. And what's interesting here is that, okay, delayed interviews are subject to memory bias, but quite a high number of reports included references to the research setting. So are these references a sign of connected consciousness? And we tried to look at this by utilizing a serial awakening paradigm during experimental anesthesia, so that in stepwise increments, we rendered our participants unresponsive. While they were unresponsive, they were presented event-related potential stimuli, sentences to look at the N400 event-related potential, and also they were presented with emotional stimuli, because emotional and semantic stimuli are differentially processed in the brain. And then while still continuing the infusion with a constant dose, we woke them up or tried to woke them up and interviewed them about their subjective experiences. And if with the same dose the participants became unresponsive, again, we repeated the procedure. Then we increased the anesthetic dose by 150%, had the same procedure, except now we interviewed them after a recovery phase, because of course they could not be immediately aroused from this deeper unresponsiveness. And what we found was that more than 80% of our interviews included experiences from the unresponsive period. And we are now pretty sure that they took place during the unresponsive period because we woke them up from under anesthesia. Most of these experiences were dreamlike hallucinatory experiences, and a very high number of the reports included memory incorporation of the experimental setting. There were a few reports that indicated being connected. So there were reports that the participants were aware of what had been going on. But when we checked the logs and the EEG tracings, we noticed that these participants actually had a spontaneous arousal. So they were in that sense not fully under anesthesia. And the emotional stimuli that we presented to the participants during unresponsiveness, none of our participants spontaneously reported anything related to these stimuli, but when we presented them a recognition task afterwards, so stimuli that they had heard during unresponsiveness and novel stimuli, they actually recognized faster the stimuli that they had heard during unresponsiveness. So their reaction times were faster. So basically, what we hope to have shown with this study is that experiences take place during behavioral unresponsiveness induced by anesthetics. So consciousness is not abolished. And while this memory incorporation is common, it seems to be really a similar type of phenomenon than what we see in a sleep lab on the first night. So when participants sleep in the sleep lab the first time, they tend to report a lot of dreams with reference to the novel environment. And so being anesthetized in a novel environment easily leads to memory incorporations of things that you have seen, heard, been exposed to prior to the anesthesia. But it's extremely rare, even under experimental anesthesia, to have recollections that indicate connected consciousness. And because sleep and anesthesia have been suggested to use the same or similar neural circuits and mechanisms, in our latest study, we force awakened same participants from dexmedetomidine or propofol induced unresponsiveness and non-REM sleep and interviewed them immediately afterwards, and then compared recall rates and content of these experiences. And we managed to do 76 interviews in the anesthesia experiment and 73 in the sleep experiment. And the experiment was complicated by the fact that we actually did simultaneous PET imaging and EEG recording. So the number of awakenings we could do was limited. And about 70% of reports from anesthesia and 65 from sleep included experiences. So there was basically no difference in the recall rate between sleep and non-REM sleep and anesthesia. And also there were no differences in the types of experiences that our participants reported. Most of the experiences were these dreamlike hallucinatory experiences. And again, memory incorporation of the research setting was really common. Of course, a novel environment of sleeping in the PET scanner and being anesthetized in a PET scanner might have been the cause for such high level of memory incorporation. So basically, what we hope this study showed is that when we interview the same participants, anesthetic-induced unresponsiveness and early night non-REM sleep are phenomenologically similar, and they are characterized by disconnected conscious experiences with about corresponding recall frequencies and content. And now I've used up all my time. I had one more slide that showed the PET results from the same study. And we can come back to these during the discussion so that we also have time for Rob's presentation. And if anyone has question about these PET results, we can maybe go through them during the discussion. So thanks for your attention, and also thanks for the whole team that I've had the privilege to be part of in these studies.
Thank you, Katja. That was very interesting. I think the way that this is normally run is that we have some questions now, and then we can continue and then have a general discussion afterwards following Rob Sanders. But is that how you would like to do it now?
Yeah, I think Rob had a comment or question in chat.
Yeah, so I'll just jump in. So thank you very much for inviting me. For those that don't know, I'm the Nuffield Chair of Anesthetics at the University of Sydney. And the work that I will present in a minute was work done at the University of Wisconsin when I was there. It was just a comment on the Sarasso paper. And, you know, it's a phenomenal paper. really excellent work. But the methodology is a bit different to the sort of elegant methodology that Katja describes. So they sort of asked people after, the anesthesia was completely finished, do they recall? And there was quite a long gap in some patients between when they asked. I think that gap in some patients or some participants, was months. So I think that we need to, I think that the data that Katja presented are really, they're the cutting edge data in the field and should be respected as such. So I think, Katja, you probably done yourself a disservice. And also, Katja, if you wanted to describe your PET results, I was thoroughly enjoying your talk, so I'm very happy for you to carry on. But I can speak too if you prefer.
Yeah, but I know that your talk will actually tap more into the kind of brain mechanisms of anesthesia and consciousness. So we can come back to these PET results if anyone's interested. They've been published, so they are not they are accessible.
Okay, I have an immediate question for your talk. So the definitive argument for the dream content not to be an instantaneous confabulation, right, is that you provide some kind of stimuli during the anesthesia or during the sleep that somehow is part of the narrative. But it doesn't seem to me that evidence is very convincing. Or did I not understand that? And then what would be an alternative way of doing an experiment to make sure that it isn't something that's confabulated in a second, the moment you wake up?
So in dream research, there is a long-standing debate about whether dreams really do take place during sleep or whether we always confabulate our dreams on the moment of awakening. And without going into that discussion, the reason why we presented these emotional stimuli during the unresponsive period was basically because emotional stimuli is relatively arousing, and it's been shown previously that emotional stimuli more easily leaves a mark in your memory than, for example, semantic stimuli. So we wanted to check whether or not people would actually be connected enough to report anything related to the stimulus, because it was quite unpleasant and salient. And none of them spontaneously recalled or reported anything about that. That's not in itself surprising, because there are a lot of studies on sleep about how we incorporate external stimuli into our dreams, and the incorporation rates are quite low, except for painful stimuli. That's the kind of stimuli that most easily gets incorporated into our dreams. So of course, we didn't want to use painful stimuli with our study participants. But what I found surprising was that, in fact, in both groups, those who got dexmedetomidine and propofol, both groups had shorter reaction times to familiar stimuli. So stimuli they had heard already once, and we only played them once during anesthesia, suggesting that there was implicit memory formation. And in fact, in the dexmedetomidine group, they also responded yes to recognizing these stimuli that they had already heard, but not in the propofol group. So I hope that answers your question.
Yeah, that was very interesting. I have other questions, but maybe let's open it up to anybody else who has a question for Katja.
If nobody else has, then I'll have a quick question. Maybe it also relates to Bruno's question and also something that Rob is going to present. But is there a drug that can potentially protect people from forgetting? So that you can, if you administer that in addition to propofol or something, you remember the dream much, much better and for a longer time.
Not as far as I know of. That kind of a drug would be a really cool agent for a dream researcher. But of course, in anesthesia, it would be possible to use a stimulant like physostigmine to arouse the participants quickly from unresponsiveness. But that is quite a shock to the participants. So if they would have a deeper anesthesia and be quickly aroused from it with a drug, I'm not sure if that would actually help recall or not. Probably make them really confused. Rob might have an answer to this, because I'm not an expert in anesthesia.
There was a finding of the drug that Ben Baird tested to induce the lucid dreaming more often, right?
You're talking about galantamine, which is a nutritional supplement that increases acetylcholine in the brain and slightly increases our REM sleep amount, just very slightly. It's used as a mainly as a medication for Alzheimer's disease. And because it increases acetylcholine in the brain, there's been a suggestion that maybe it could, and it intensifies REM sleep, it could induce lucid dreams, but using that in the context of normal dreaming or anesthesia, I wouldn't see any point. It could maybe intensify our dreams or increase recall, but not necessarily. And most people who take it with a sufficient dose tend to get gastrointestinal symptoms, so, or at least some.
Yeah, there's some comments. I think we can all see them. I'm not really sure what Ariel is referring to.
I'm just speculating, but I'm not an anesthetics expert by any means, so I was just wondering if anyone else knew more about it. But it's probably, my speculation's probably not worth even discussing. We can move on.
Well, it's possible, but we don't have any positive modulators of the NMDA receptor that are clinically available to my knowledge. If you know of them, then...
No, I've played with a few back when I did animal work, but yeah, I don't think they're available for human therapeutic purposes. So that's why I was curious, but thanks.
I can maybe make the notion that typically what's reported with ketamine is that people report these strong visual hallucinations and long vivid dreams, detachment from their own body or their personality. And yes, but usually these reports are actually acquired quite a while after the anesthetic has dissipated. And in another study, we used ketamine on healthy participants, and we tried to interview them immediately after terminating the infusion, and it turned out to be almost impossible. So even those who were responsive, so they seemed to be awake, they were in a completely dissociated state. So I'm not sure if they understood the question. Those who understood the question had really difficulties expressing themselves. They had just, motor dysarthria. So the ketamine reports, it would be really cool if we would have some agent that could get the ketamine participants talking faster or sooner after the original experience. I'm sure that the delayed reports that we get from them are reflective of the types of experiences that they had, but it would be really wonderful to get immediate reports out of participants who are receiving ketamine. Of course, this is also dose dependent.
I was just going to talk about that actually, or ask you, like everything, it's all about the dose, right? So when you are when you're doing these experiments, clearly there's going to be a sedative dose and then an anesthetic, more of a surgical dose. And there's different potential target mechanisms involved. The sedative dose being GABA receptors, the anesthetic surgical dose maybe involving a whole bunch of other targets that have nothing to do with sleep. How do you separate those effects? Are you really looking at that lower dose, or are you actually looking at surgical concentrations when you report your results?
There is also a lot of individual variation how susceptible people are to different drugs. So in fact, in the first studies, we used an individually titrated dose with gradual increments until the person lost responsiveness. And there could actually be quite huge variation. So some of the participants actually got doses that are clinically relevant, so close to what they would have been getting in if undergoing a surgery, of course, in conjunction with other drugs, while some of our participants were really rendered as unresponsive with the first couple of doses. So in most of our studies, the doses are actually individual. In the last study that I didn't, in one of our latest studies that I didn't mention here, it was the same where we did the ketamine interviews, is that we gave them an identical dose of dexmedetomidine, propofol, sevoflurane, or ketamine. with the idea that about 50% of our participants would remain responsive with that dose, and another 50% would become unresponsive. But in fact, what we noticed is that, okay, we had few participants that were unresponsive throughout the experiment, some who were responsive, but most of them fluctuated between being responsive and unresponsive. So it's there's also the time effect of how the drug is metabolized. And again, I would say that Rob might be a smarter person to answer these questions that relate to anesthetics and doses.
I might make a couple of comments, but I agree with everything you've said, Katja. So we have had the same experience with ketamine, and I don't know what state high dose ketamine is, and I don't know if there's many anesthetists in the world who could describe it, but participants can't interact with us in that very high dose state, and we can't get information from them. We've even done painful stimulation during them to try and wake them up, and we can get limited interaction with them at high doses, but we certainly aren't getting descriptions from them. And therefore, I don't know whether all the dream reports actually correspond much more to sort of non-anesthetic doses of ketamine, which would be associated with NMDA receptor antagonism rather than blockade of other channels. So I think ketamine is a very, very hard drug to understand, and we've probably oversimplified what the ketamine state is in our wish to put it in a pigeonhole. We've done similar individualized drug titration. We're more interested in the conscious state than the actual drug concentration. But I would say that we think that what we're doing is relevant to the drug doses that can be used in the operating theatre. It's impossible to know because obviously in these settings, we're not doing an operation. And so there's not that counterbalancing noxious stimulus that's trying to wake the patient up as we're trying to put them to sleep or make them unresponsive or change their conscious state. So I do think it's clinically relevant what we're finding, and I'll talk a little bit about what the clinical work we've done that Katja's touched on as part of my talk.
Well, maybe we should go ahead then and have a listen to your talk, Rob.
Sure.
And we can have a more general discussion with later as well, and get back to Katja's PET results, I think, which were probably worth another look.
Yeah, definitely.
Yeah, so we'll just keep moving then, if that's okay with everybody. And I've looked at your CV a bit. It's very interesting trajectory. A bit of Imperial College, close to some of the leaders in the field there, I guess. And then the move to Wisconsin with Giulio Tononi. And I think you're one of the few people who are spanning these clinical kind of explanations to also just basic research with connectivity in the brain and what happens under anesthesia. I did note that you both have a George Mashur number of 1 in connection to each other. So you both published with some of the same people. And I think that speaks to just really trying to understand, beyond the clinic, what actually, just the question of consciousness, which is of interest to all of us and to this group here. So go ahead and Rob, tell us about your story, and then we'll have a nice discussion after that.
Okay, great. Oh yeah, I think I need permission to share screen actually. Have I got that now? Oh, I've got it, sorry. Okay, so start by just acknowledging that this is a collaborative project. I'm probably going to try and take all of the credit for myself, but I shouldn't. And I'll describe some of the people who've done all the hard work at the end. I'm really going to just focus on the 10-15 minutes that I've got, focusing on our recent paper on this, which builds on the work that Katja has done and Harry Scheinin and the whole group there with the serial awakening paradigm under anesthesia. I moved to Wisconsin to work with Giulio Tononi, and they were doing serial awakening in sleep. So we wanted to try and replicate that with similar motivation to Katja under anesthesia and understand if we can really extrapolate between the states, as an anaesthetist, I don't want to wait for anyone to go to sleep as well. I need to take control of that situation. So it's much easier for me to give a drug than to be patient. I don't have any financial relationships to disclose. I do have an academic relationship with Katja that we're working on. So some of the work which I'm going to present now, we hope to test in Katja's and Harry Scheinen's data to see if really this is generalizable, what we found, or this is something unique to our data set. And you've already heard about this sort of these phenomenal descriptions of the conscious state. So we like to describe consciousness as the ability to, or the actual subjective experience that people are having. Connectedness, whether that subjective experience is related to the external world, whether sensory stimuli is driving that subjective experience, and responsiveness, whether people will then interact with the external world. And obviously the serial awakening paradigms have been instrumental in trying to bypass the need to always use the responsiveness as a surrogate for conscious state. And I won't labor this because Katja's been through it, but you can use different sleep stages to exemplify those potential interactions. And obviously Katja's already described why this is an oversimplification, and that consciousness can occur, an absence of consciousness can potentially occur in each of these sleep stages as well. So the objectives I really wanted to talk about was describe a bit about why we're interested in sensory disconnection, specifically from a clinical perspective, and also touch on where we're at in the clinical world and why we need these sorts of serial awakening studies and what we think they'll bring. We're going to explore some potential markers of the conscious state using the EEG, and obviously that'll be a useful segue back to Katja's PET data. And hopefully there's some mechanistic insights, and I really would be excited to talk more about collaborations about trying to understand mechanisms that lead on from this, because there's only a certain amount of mechanistic manipulation we can do in the human species, and there may be other species where we'll be more suited for doing research to understand the fundamental mechanisms. But specifically, I'll describe that mechanistic the mechanism data, that with limited mechanistic data we have, so that we think provides correlates, neural correlates for unconsciousness and sensory disconnection under different anesthetic drugs. Specifically around sensory disconnection, my view is that dreaming is an acceptable state of anesthesia, provided my patient, when I anesthetize them, is not aware of surgery, they're not aware of the external world, then I'm happy that I've achieved my role as an anaesthetist. Some of my colleagues would say, as long as they don't remember it, that's all that matters. They're the amnesiologists. I still believe that I'm sort of maybe I should be a dreamologist, but someone who the anesthesia provider, so you're not aware, you're anesthetized to the surgery. I don't mind if you're having a dream that is entirely based on your internal world that you construct internally and don't get sensory information from the external world. Interestingly, if you accept that view, titration of anesthesia to unconsciousness could be an unnecessary deepening of anesthesia if there's a dose response with anesthesia and conscious state, such that dreaming is a lighter anesthetic state than unconsciousness. We may target unconsciousness because we want a buffer, but it could also be an unnecessary deepening, which is an interesting thought experiment. We have some data on the dose responsive effect of drugs and drug concentrations and those conscious states. And more fundamentally, I think that there are great mechanistic insights that we can see from anesthetic drugs, not just about molecular mechanisms, but also about the neural correlates that can give us insights that may be relevant for other disorders of consciousness and sleep itself. And you've seen these wonderful data that Katja presented, so I'm not going to labor this, but what we know is that sensory disconnection occurs readily under sedative doses of drugs, and we think, and I'll present some data on that there's a bit of a dose response, such that the higher dose you'll get, the less likely you are to get a dream state if you were to wake the patient up, rather than an unconscious state. But dreaming occurs with clinical relevant doses of the drugs. But what happens in the operating theatre, and Katja presented some of these data, but when patients report after surgery that they remember surgical events when they thought they were under general anesthesia, is a very, very rare event. So that occurs in about 0.1, about 1 in 1,000 to 2 in 1,000 patients. So it's pretty rare. But when people can report intraoperative events at the time but don't necessarily have explicit memory of those events after surgery, is much more common. And we've seen that after intubation, for example, when we ask people with the isolated forearm technique to squeeze our hand as a behavioral measure of consciousness, that about 5% of people wake up. And in young adults, we've got a paper coming out shortly that shows that it's about 11%. So even with standard clinical care using present standards, about 1 in 10 patients may wake up after intubation and report that they were conscious. These patients won't remember it afterwards, and we can argue philosophically as to whether this is important if you don't remember it. But it definitely occurs, and connected consciousness events are occurring much more frequently than the most of us think they will. And I also have put up Lenassi's meta-analysis here. This is an inflated number suggesting that 35% of people may have episodes of connected consciousness or wakefulness during standard clinical care, because it's included studies where, for example, anesthesia was titrated down to get a response out of patients. So it's a useful potential addition to the literature, but basically it's an exaggeration of the state of affairs. And I think sort of 10 to 20% is a more reasonable estimate, but still, I think many people would still find that alarming. And in the operating theatre, we use different depth of anesthesia monitors to try and track brain activity. And these work with variable success, but none of them detect these episodes of intraoperative connected consciousness with great reliability. And we've done a study on this. And one of the studies we embarked upon doing was to test whether this measure, which is merely this emergence of this frontal alpha delta oscillation in the EEG with the induction of propofol and general anesthesia, whether that was truly a marker of unconsciousness as proposed by Patrick Purdon and Emery Brown from Harvard. And this is a spectrogram with time after drug concentration on the x-axis and the frequency on the y-axis of the EEG. And you can see with this large bolus of propofol here to this volunteer, there was a delta pattern emerged, and then immediately afterwards you got this alpha oscillation co-occurring. And this co-occurring alpha delta signature has been proposed as this measure of unconsciousness. And what we wanted to do was to test whether this measure and various other measures as well were associated with unconsciousness under anesthesia, and it could be a reliable marker. And to cut a long story short, work with Amy Gaskell in this multi-center study basically showed that this frontal alpha delta EEG pattern is not a signature that you can rely on to detect responsiveness under anesthesia. And this is the data from a single subject showing a similar spectrogram with time on the x-axis, frequency on the y-axis. You can see after propofol was induced about here, there's an emergence of the delta oscillation. Volatile anesthesia was used in this case as well. And then so there's a slightly slower emergence of the alpha oscillation, and the patient was intubated about here. The patient was then asked to squeeze a researcher's hand at this point after intubation, and at this point after intubation, and responded positively, and at this blue line responded negatively. But not a convincing change in that the alpha delta oscillatory pattern has changed during that time. So a few questions here. Is this just the wrong pattern of activity? Are there other patterns of activity we should also have measured? Is a single frontal electrode sufficient to really capture consciousness in the brain? Do we need to do more? So those are the sort of motivating questions which suggest that we need to be doing serial awakening studies, because we need to get a much more profound understanding of the brain and the physiology that's underlying consciousness in these different anesthetic states, to then be able to go back to the operating theatre and actually monitor brain activity in a meaningful way that patients want, because patients want in the main to not be aware of surgery. So we went back to the drawing board following similar methodology that Katja has presented with serial awakening. We did a volunteer study with 20 subjects with dexmedetomidine, 6 with propofol, and 15 with sleep. We've also done 15 subjects with ketamine that I'm not including here, partly because it's a hard state to quantify in the same way. And I don't think necessarily you can just merge ketamine with these other states, because propofol and dexmedetomidine produce a phenomenologically similar state. Ketamine is producing a very wildly different state, and wildly different between individuals. The study was planned to be slightly larger than it was, but unfortunately we had to stop it because I moved to Australia to escape COVID, not expecting COVID to follow me. I didn't bring it, by the way, but it did follow to Australia. But nonetheless, we pushed on and analyzed the data we had, where the sedation anesthesia was titrated to achieve these different states. Subjects were aroused frequently, about every 5 minutes during sedation over 4 hours, to probe their conscious state, and were asked a series of structured questions to elicit what their conscious experience state was immediately before awakening. We did this with high density EEG data collection with 256 channels, and we calculated power in a series of different bands, which are fairly standard in the EEG literature. And that's just been published and it's out online now presently. I'm not going to go through this for the interest of time, but I will mention some of this stuff because I think it is relevant to the conversation we were just having. So you can see here in panel A, roughly the schematic which we followed, which is very similar to Katja's data in the Radek paper, where we, for example, with dexmedetomidine, we'd have these two standardized drug steps, and patients would oscillate between different conscious states at these two different steps, with slightly more unconsciousness in step 2 and slightly less unconsciousness in step 1. We also did similar titration with propofol. In reality, propofol, because it's a more potent drug, required a little bit more titration than these two simple steps, but everything was done with collection of drug concentration levels, so or predicted drug concentration levels anyway. So we can include drug concentration as a covariate on analyses, and that's important for us because we're not interested in studying non-specific effects of drug. We want to drill down what are the specific neural correlates of each conscious state, which we can detect under dexmedetomidine, sleep, and propofol. These are the number of wake-ups that we had in each state. Now, the wake data in red were collected before drug was started. You'll note that there's no orange segment in this pie chart under sleep, because the orange segment is essentially the same as wakefulness with drug on board. We call it connected consciousness to differentiate it from wakefulness, because there is drug exposure in the propofol and the dexmedetomidine group. We had about 72 wake-ups, for example, in the wake state with dexmedetomidine, 24 in propofol, and 65 in sleep. You can see that with dexmedetomidine and sleep, similar to the data Katja proposed, there's a lot more dreaming than there was unconsciousness, and that might be, Katja could probably advise me as to whether we didn't capture enough unconsciousness because maybe we were waking everyone up too much. But we can talk about that offline. And with propofol, much fewer wake-ups than with dexmedetomidine because we only had 6 subjects in that group. You can see how these wake-ups and conscious state varied with plasma concentration for dexmedetomidine and for propofol, and you can see you get that inkling that connected consciousness occurs at lighter drug concentrations than unconsciousness, for example, at those deeper concentrations. But certainly 4 mics per mil plasma concentration of propofol is very much in the clinical range for drug concentrations for propofol. I will point out with propofol, it's slightly difficult to get a lot of reports of these higher concentrations, because sometimes when you try and interact with the patient, I couldn't understand anyway what they were saying because of the slurred, garbled speech they'd have. So that certainly made capturing data at the higher concentrations more difficult. Briefly, because I know I probably already ran out of time, these are what the EEG spectra look like for a frontal electrode, parietal electrode, and occipital electrode for dexmedetomidine, propofol, and sleep. What you'll notice is that for dexmedetomidine, for example, the orange and red lines are pretty similar, and then the purple and blue lines are pretty similar. So orange and red are essentially wakefulness, with red wakefulness, no drug, orange connected consciousness with a drug. And you'll see that the blue and the purple lines are much more similar. That's disconnection in blue and unconsciousness in purple. But there's no obvious signals suggesting that you can easily differentiate across states with just the power spectra here. And there's plenty more that needs to be done to drill down within the power spectra if there are other signals, and I'll describe some of the work we did on that. But it's not like we can choose a single electrode and just identify the conscious state from that, though we tried. And the reason that we tried is that originally when we set up the study, we were inspired by this notion of the posterior hot zone, that there would be an increase in low frequency or delta power over occipital regions, and that would correlate with conscious state. Now, so this was our a priori hypothesis, which we had. And what we tested was whether wake, connected consciousness, disconnected consciousness, or unconsciousness showed an escalating change in delta power across conscious state. And in fact, we found that, much to our sort of pleased surprise, that with dexmedetomidine, at least, that did seem to hold, that the amount of low frequency activity over the posterior regions of the brain reflected conscious state, at least at a group level. Now, in panel D, you'll see the receiver operating curve, where we try to classify, based on the dexmedetomidine finding, the delta power that would differentiate conscious state in sleep and propofol. And while delta power could differentiate conscious state in dexmedetomidine, it didn't work when we looked at a combined group of sleep and propofol. So this didn't seem to be a generalizable signature of the conscious state that could work across drugs and sleep states. I will also say that we did a control for drug concentration in these linear mixed effects models, because we didn't want to just be studying non-specific effects of drugs, given that there's collinearity between drug concentration and change in conscious state. We wanted something that was specific to the conscious state.
Rob, sorry, just for the clarification question.
Mm-hmm.
Is the previous AUC curve, is it about within subject discrimination or across subjects discrimination?
This is across subject discrimination.
Okay, across. So that's very difficult.
Yeah. Across subject, across state.
Okay.
We wanted perfection now. So we sort of went back to the drawing board, and we were inspired by Francesca Ciclari's paper in Nature Neuroscience with Ben Baird and Giulio Tononi. We did source reconstruction using E-Loretta and looked at different power bands of activity, and also looked at this beta delta ratio that they'd been very keen on, also the gamma delta ratio, which they suggested could differentiate dream states from unconsciousness. So we looked at each of those power bands and those ratios, and those estimates of power are plotted here in the left two columns. I just got to move the bar here so I get it right. For connected consciousness on the left and disconnected consciousness on the right. And then in the middle column, you see the T threshold of maps showing statistical differences between disconnected consciousness and connected consciousness. And the orange shows a positive difference, so there's more power in the disconnected consciousness, and the blue shows a negative difference, where there's more power in the connected conscious state. Now, the important take-home message from these multiple tests was that there were multiple power bands that could differentiate the disconnected conscious state from the conscious state, from the connected conscious state. And this is controlled for plasma concentrations again. So it seemed to be that there were multiple mechanisms for sensory disconnection occurring, including increased slowing. So this is 0.5 to 1 hertz in a very slow oscillatory band over posterior parietal regions, similar for delta and theta. So it's increased slowing posteriorly with some frontal involvement, but also loss of high frequency activity. And that's probably most evident when you look at the beta delta ratio. You can see changes all over lateral aspects of cortex. We then did some machine learning models, which I'm not going to go into in great detail because of time, but we were able to show that when you did the same, analyzed the same data for dexmedetomidine with a machine learning model, you could nearly get 100% classification accuracy. We then tested that SVM-based machine learning model on sleep and propofol data, and we could classify above chance using these power metrics, the conscious state in sleep and propofol, based on the metrics we obtained from dexmedetomidine. So we were hoping we're kind of gradually edging towards a closer signature of the disconnected conscious state. But the classification accuracy here is about 74%. So it's certainly not perfect, and we can certainly try and refine that further in future studies. Yeah. I've nearly finished with the shiny brains, but this is where we try to now distinguish between disconnected consciousness and unconsciousness. And so these are the power views on the medial and the lateral sides of the brain, for disconnected consciousness on the left and unconsciousness on the right. And then there's a T map next to it between the unconscious state and the dream state on the right. And you can see that, and we tested all of the same bands as on the prior slide, but the only metric we had which showed statistical differences between unconsciousness and disconnection was this beta over delta power ratio. And it's the same ratio that Francesca Ciclari and Ben Baird sort of studied in their paper with dreaming. So that kind of shows that there is a precedent in the area for looking at this ratio. And what we found was really interesting, because what we found was that there are these hubs in anterior and posterior cingulate bilaterally, where there are differences in the beta delta ratio when subjects report no experience when awoken from sleep versus a dream state. There's also a difference in this lateral parietal area, and as I'll describe in a minute, that's only seemingly relevant in our hands in the anesthetized state, but didn't seem to pertain or work so well in the sleep state. But before I go on to that, I'll just show you an inset image here of what Francesca, Ben Baird, Giulio Tononi showed in their paper, is that there was this posterior sort of temporal hot zone that was important for distinguishing dream states just under sleep. But there isn't such an obvious anterior cingulate hub, and this has obviously been a strong side of strong evidence for a posterior hot zone, understanding a sort of minimal neural set of consciousness that would support dreaming. And in our hands, we also have this anterior cingulate hub. So we then looked at the effect estimates once we've adjusted for drug concentration where necessary in the anesthetized states, for each of those areas across the state, so dexmedetomidine, propofol, and sleep. And you can see that these anterior regions are similar across in dexmedetomidine, propofol, and sleep when you look at the effect estimate from the models, both on the left and the right. What's interesting is that the parietal hub region here in green wasn't significantly associated in sleep, but was associated in the anesthetized state. And when we used all 5 regions to try and create a classifier based purely on the beta delta ratio, we did actually get better than chance classification, but still the classification was weak. It's only about 62%. So there's some interesting, I think, future directions from this work about understanding how much of these cingulate regions are just acting as hubs, communicating variable information from other regions, and how much they are the sort of core nodes of driving a conscious experience. But certainly some evidence that you can at least identify neural correlates of conscious experience that can be used to classify across subjects and across states, which is a pretty high bar, I think. All right, that's enough from me. I'll just say that in the future, I'd love to come back and talk a bit about the work that Jordan Wehrman's doing with me in my lab on further markers on sensory stimulation, on sensory disconnection using evoked stimuli, and how we're going to try and we need to start moving back to the clinical arena, I think. But certainly looking forward to more collaboration and discussion as part of this to work out how we can drill down mechanisms more thoroughly.
Thank you, Rob.
Thank you.
All right, let's just open it up for discussion now, if there are any questions. I'll start with a first question. So I did note that that you have a lot of work done on delirium, especially in older patients.
Yeah.
What do you think there's a connection, if any, between the propensity of older patients to have delirium issues after anesthesia and some of the mechanisms you might be discussing today with regards to a link to dream sleep? Why would that get worse with age?
Well, there's a lot of levels to that question, Bruno. So the first is whether anesthesia is associated with delirium, and bizarrely, anesthesia is not the primary cause of delirium. No doubt, when waking any patient up from anesthesia, if you were to ask them at the right point, you could identify a delirious state. So everyone is going through some form of delirium on waking up, but it's not necessarily the same delirium that we talk about in our studies. That said, we have proposed a link that it's theoretically, I think, delirium could be a state where you're abnormally connected to your environment. In delirium, you have this increase in these slow frequency oscillations that occur mostly over occipital regions as well. Now, it's not clear to me that that's not just because it's easier to measure them there then there's something specifically happening there. But nonetheless, there's this slowing in the EEG, and there's an interaction to the external world, which obviously is a series of events which is carefully controlled in sleep, where the EEG slows, the brain activity changes, and the body has worked out it's probably best to be unresponsive at this juncture. Presumably there's a good reason for that. So delirium could be conceptualized as an abnormal state of sensory disconnection, or an abnormal level of connectedness to the environment. Now, how we would manipulate that and whether we should are two different questions that we need to sort of engage with. But certainly the links between sleep deprivation and cognitive change, and in that sleep deprivation state that people are also are predisposed to delirium, those sorts of factors. I think there is a big overlap here between understanding what's changing at a synaptic level, what's changing at a cognitive level, and then what changes with age, which is then superimposed on that, and whether that's just a cognitive reserve element, or whether that's that patients are more easily connect to their environment as you get older. I don't know the answer to that, but those are all good sort of questions that need to be answered.
Yeah, thank you. Can I follow up with another question?
Or maybe I have some question. Also, in relation to your work, Bruno, I think some of the work in animals, especially, has been kind of, implying different networks are responsible for different aspects of the arousal, in particular, probably sleep and also anesthesia differently, probably. Is there a possibility to manipulate these areas of the brain or connectivity by some way? I asked already drug kind of possibility after Katja's presentation, but something like ultrasound stimulation, or more traditional technique of TMS or something to wake up the particular parts of the brain to recover some aspect of the cognitive or sensory function during the anesthesia. Do you know any work or possibility of that kind of thing, either in animal or human, to understand better about the connected consciousness, the consciousness during this anesthesia?
Yeah, I mean, the only thing I can think of is there's some work being done in basically turning on protein expression during memory formation, where you can see an engram, you can see like the imprint in a way of a memory in the brain post facto as a kind of as a fluorescent readout. And that there are some tricks now that you can use in mouse studies or in fly studies, where you can actually, in a way, see the evidence of a memory in the brain. And this involves actually gene transcription that happens that relates to memory formation, and then actually putting together the circuits that were involved at that moment for that memory formation. So this is beginning to happen in the field. Not everybody believes it, but it seems to me one way of potentially finding a trace of a memory that could just as well happen under anesthesia as during sleep or other states of a disconnected consciousness. So that is one possibility, using these new kind of genetic tools. Apart from that, I'm not really sure how you would find any evidence that would be divorced from behavior, right? An animal has to show behaviorally that it remembers something. In the same way that, in Katja's experiments, there has to be some kind of way of having some kind of behavioral readout. And I think Rob's a kind of a image there where you see like what looks like no change in the spectrogram at all. And clearly different behavioral responses is a bit disconcerting.
Well, I think that the behavioral readout then, as I understand it, in Katja's work and my work, is the actual report itself. And I love this idea that you could have an imprint in the engram of recent or prior activity. What's the half-life of that activity? Like how far back in time can you trace?
I'm not sure, but this is work that's done by what's her name?
Negawa? No?
Michael, who gave a talk last week at QBI? Or Matt?
Lucy Palmer.
Oh, don't worry, it's not a test. If someone could let me know, I just think it's fascinating. Yeah. But I
Lucy
love.
Palmer is working on this at the Florey.
I would be cool if we could combine that with Katja's, evidence that you more recently incorporate, if you your new environment into your dream, right? You could put them in a new environment and then let them learn their new environment, sort of anesthetize them for longer than the half-life of the engram, and basically find out if the engram was still present at a time that shouldn't still be present if it was. Oh, you'd have to do it the other way around, actually. I need to think about it, but it's a cool idea. We need to leverage that.
It's one way to answer Nell's question, I think, in animal models. Yeah.
I mean, Yuri Salmon's waking non-human primates up with electrical stimulation of the thalamus and things. You just can't get them to communicate yet that they're having a dream. So there's ways of doing it. It's just we need to have some sort of physiological readout of what a dream state is.
Rather than physiological or behavior readout, I vaguely remember that the Adrian Owens group has also tried to do this imagery-based neural kind of communication flow. So that Rob, you said that after immediately awakening after propofol or ketamine, people are very difficult to sort of express what they are thinking, right, by language. But still, if they can understand the instruction, maybe they can manipulate the brain state by imagining one thing or the other. Would that be a possible readout for the presence or absence of consciousness?
I think it'd be a really hard experiment to do practically. I mean, the other one, for one thing, with fMRI, which is obviously their technique, the patients are not going to lie nice and still. It's got to be a relatively stable state for minutes with fMRI. It's just, that would be a real challenge. It's not a bad idea. It's just, it would be technically challenging, I think, to do it. Our approach of just throwing out the trials we can't understand or get a clear readout of is suboptimal. So we definitely need some other way of doing that. But at present, in the first stage, we just say, well, can we classify the stuff we know across states and across people, which is a pretty big task. Then we want to start classifying what we don't know. I mean, that would be even harder. So, these are big challenges.
Yeah.
And I think the drug manipulation stuff is kind of interesting. I mean, that's obviously a lot of animal work has gone into giving, they've done these arousal experiments under anesthesia, and a lot of them, once these circuits are a circuit, so once you stimulate one point in the circuit, you sort of end up with reverberant activity, which is changing the whole dynamic of what you think you're studying.
I see.
Katja?
Yeah, I also had the point that doing fMRI with anesthesia is a bit tricky. We did that with 10 participants who got dexmedetomidine in the scanner, and it included some technical innovations and also a kind of a communication problem between anesthesiologist who had to stay with the participant and another one who was doing the administration. And also, I think the big difference might be that let's assume that these participants in Owen studies who were in vegetative state, they had the motivation to do the task, that they not only understood the instructions, they were capable of doing the task, but they were motivated to do it. And I think one problem with sleep and anesthesia is that when you are really groggy, even if you hear and understand a request, you just, sometimes couldn't care less of complying to it. So there's also this aspect that we really can't control, and participants that they are not really willing to admit that, yeah, I heard something, but I just didn't bother.
I see.
I think it's interesting, Rob, that you're gravitating towards predictive processing and predictive coding, because I think that's exactly where some answers, and I think Andrion has done some work along those lines too, potentially, right now, Thomas Andrion, where, you know, the kind of stimuli that you're presenting during anesthesia or sleep, depending on how surprising they are, like if there's oddball events or, you know, just your prediction about what happens next, is maybe a better indicator than any behavior, and much more applicable to animal models.
Yeah, I mean, that is a motivation. I'll agree with the latter statement, Bruno. It's right on the money. I mean, you know, we've got this overarching idea that sensory disconnection involves a loss of predictive processing, right? If you're not updating your internal model with external sensory information, whatever sounds or whatever we play to you peripherally should be completely, you know, irrelevant, because you're not updating your dream. Now, the interesting time will come when we have enough data of people who've heard the beeps in their dream, and we can say, well, you have updated your dream. So I just don't think we've got enough data on that, unfortunately, from what we have so far. But the idea is to test that, whether you know, there's incorporation of prediction errors from the external world and model updating when we are in this so-called disconnected state. The challenge with those data are that, you know, we collect those data over minutes. While I didn't tell you this, sorry, apologies, but the EEG data, power spectrum data I'm presenting, that's from 20 seconds immediately before wake up. So we wanted to know how, you know, immediately before wake up, what does the power spectrum look like, and how does that correlate with the conscious state you report? And there's definitely a change that you can trace back in time. So for up to a minute before, you know, the same classification models work. We haven't published those data, but we've done that test. But it's the further out from a minute you get, the weaker the classification performance and the more noise you have in the model. And then, so to collect enough evoked potentials, you know, we're collecting data over several minutes. That in that time period, conscious state likely changes or could change. And so there's a little bit more ambiguity on those signals. But if we can get confident in them, and we've got some, we think we can, we just got it, there's a bit of faith, then we will have something we can translate into animal models for mechanistic studies.
Yeah, I mean, one could imagine that, you know, that consciousness, just to, you know, to speak broadly, you know, and following from Anil Seth's kind of view of a, you know, a controlled hallucination, is just really that, you know, that model of the world that has to be either, you know, validated or you know, updated, and that whether it's anesthesia or, you know, or dreaming. And I kind of liked how you started there, Rob, that it, in a way, it doesn't really matter. You could be a dream, what do you say, a dream esthetist or something.
Yeah.
And in a way, that's where anesthesia started, right? So like the first anesthesiologists were mesmerists,
right?
Were actually hypnotists. And that's how the first anesthesia operations were done. And then it devolved into quackery, right? But until they discovered that the new American drug, which was ether, they were heading in the direction of hypnotism as a potential way of doing surgery. And then that lasted a couple of years, and then we went to actual substances that could be much better controlled and applied to everybody. But it's interesting that there's that common history in a way that might have a common explanation.
Yeah. Well, yes, I suspect there is overlap there. And, you know, the reason that I think predictive coding is important for someone like me is it gives an established framework in which to start testing some hypotheses. I mean, the other thing I like about studying sensory disconnection is that there's some form of report to confirm that the patient was experiencing something, but not the sensory stimulus from the external world, right? The problem with unconsciousness, Katja brought up, which is that there's no behavioral definition for unconsciousness, right? So there's no way I can really be confident someone can wake up and say, absolutely, I was unconscious in that time period, because of the conflation with memory and other issues. So disconnection is a much more tractable scientific question. I think it's relevant to my clinical work. I think I should be a dream aesthetist. We need to work on the wording, but yeah, a dreamologist. And, you know, I think that from those perspectives, it's both scientifically tractable. We can do experiments, and then also it's going to have clinical applicability. I think it makes more sense. I think everyone still wants to boil down to what is a subjective experience and how is that formed. And I think that's a much more difficult question that I think, well, now and Bruno and everyone here is obviously very keen on that question, but it's a very, very hard one.
And it's an impossible in animals, right? So that's why we're beginning to gravitate towards predictive processing as well. Let me just introduce Dennis, postdoc in my lab. You can see his face, who's working on sequential dependencies or sequential processing. And for me, that's the only way we can study this really in an animal model.
Yeah, see, the problem is with the animal model still, you still need to give them those predictive processes from the external world. So, I mean, I would fundamentally support what you're doing, but argue it's disconnection you're studying. But, you know.
Is there anything you'd like to add, Dennis? Or Katja has her hand up. There we go. Katja?
Yeah, I had this, there is actually in dream research, there is this relatively weird theory that predictive processing occurs during dreaming, that even though there is this sensory disconnection, it's actually the function of dreaming to kind of give the system unpredictable input so that these internal models can be trained with surprising surprising information. I'm not saying that this theory is particularly testable or even plausible, but I thought that it might be of interest to hear for those of you who don't focus on dream research, that this predictive processing paradigm has kind of been taken into account also in dream theories. And it might be that this predictive processing is not only based on external stimuli, but also our brains do it also when they are only internally activated and have, internally generated subjective experiences, which would again make it really tricky to distinguish between these.
Yeah, so I mean, and just building on that, I mean, I think that's kind of key. I mean, I agree. I don't know how to study that, but it's the fact that the brain is driving its own internal prediction models rather than using external information to drive them is the kind of key thing that we'll be trying to dig down on, rather than that more fundamental central core of what the purpose of the dream is, which is, a real challenge, but really important to understand too.
Yeah, Ellie, did you want to speak on the chat out loud about your question, Ellie Matar?
Yes, no, I just was worried a bit about my background noise, but I think they were absolutely fascinating and thought-provoking talks. I just kind of am wondering, I think we're treating these sort of states of consciousness from disconnected, connected to unconsciousness as potentially categorical variables, but I mean, presumably they may also exist on a continuum, and I wonder if maybe already are, but treating them as either rank ordinal or whether there's a sort of continuous parameter that could be used as a surrogate for these states that may bring out more statistically significant relationships between, say for instance, electrophysiological variables and images and so forth.
Yeah, I think that's a great point, in Dr. Matar. The reason that we've done that is because we're simplistic physicians, as I think you may fall into the category of, and we like to categorize things as yes or no. So diagnosis, yes or no. So we went through the diagnostic element. I think that your point is is really well made when it comes to something like disconnection, because there's different levels of disconnection. So, just because someone doesn't respond to the tone we play them doesn't mean they wouldn't jump off the table if we cut them with a knife. And I also think that that intriguing situation where someone incorporates their alarm clock into their dream, but doesn't wake up from their dream, shows that there are these kind of potential situations that can arise where people have variable levels of disconnection, but we just don't have the quantity and the nuance and the data from our data set to address that. But thankfully Katja's got a hand up, and it's going to save me from saying anything else silly.
There is a lot of discussion of whether consciousness is an on-off phenomena or whether it's kind of a gradual. And I think that when we talk about the presence or absence of consciousness, there either are experiences or there are not. So it's kind of either or. But as soon as there is something, then we have this kind of a huge continuum from extremely simple experiences to full richness of the phenomenal world that we experience. So as Rob said, it's when we have these experiences, then the process becomes gradual, and we might have trouble distinguishing what it is when people are incorporating an alarm clock but not waking up and continuing to dream versus what's the difference if you just wake up to your alarm. But that's kind of my personal opinion, more than a kind of a scientifically argued stance.
Thank you.
Dennis, you have your hand up.
Yes. One was a question which has become kind of a comment now, because I think which was how to fit in the idea that external stimuli do change our dreams, and it's quite common, particularly in REM sleep, to have outside stimuli integrated into your dream, which is really interesting. It happened a lot to me personally when I was young, a lot. So it's interesting. I mean, you could probably already intuitively see that there would be two, one for NREM and one for REM, but maybe more. So that's for a comment, because, and the other thing was, if more to Rob, if you could expand on the comment that you made that if we're studying predictive coding in animals, we're really just studying disconnected states. Is that just because we can't get reports from the animals?
Well, if you can't, so if you, assuming that you're studying, say, predictive processing with anesthetic drugs, which is probably the limited about I should probably comment on that. We think, okay, that the low concentrations of anesthetic drugs, the first thing that happens typically, not in everyone, is that you become disconnected from the environment, and then the sensory information is not getting into the brain. If the sensory information isn't getting into the brain or being processed in the same way, it doesn't mean that the animal isn't in a dream state, right? Or it doesn't, and so it doesn't mean that the information is not, it means the information is not being incorporated into the conscious mechanism, it would appear, based on our human data. So, you're studying the minimum neural circuitry required to prevent incorporation of that sensory information. And I would argue that's disconnection, particularly if you're using anesthetics.
All right. Yeah.
I personally think that doesn't in any way invalidate its importance, but and would encourage you to keep doing it. It's just, and also virtually every animal paper that talks about loss of consciousness when it comes to loss of responsiveness, because of the difficulties in distinguishing those between in animals, across animals.
So it's just in general very hard to distinguish in animals disconnectedness from unconsciousness, basically, right?
I don't know of any way of doing it.
Yeah. Okay. No, but it's an interesting question in and of itself, and I hadn't thought about it.
So it's interesting. So one of the things in Katja's PET data, for example, and Katja, forgive me or correct me when I'm wrong, but they grouped unconsciousness and disconnection together because of this idea that these are sequential states, right? But we don't actually know that something flip-flops, so you kind of become unconscious, and therefore you turn on all your connectedness circuits if they are different, which we don't actually know. So, and the problem is you can't verify that because you can't get a report from the patient to say, I was unconscious but aware of the environment, right? So, so it's all entirely like that grouping makes sense based on the plasma concentrations, it makes sense on the phenomenology, it makes sense on the brain activity stuff that we see. It's not like we see, for example, when we did the contrast between disconnection and consciousness, bits of the brain turning off and other bits turning on, right? So it does seem to be reasonable to group them if you're studying disconnection. It's just you'll get a bit of additional information that from the unconscious state that isn't necessarily the same in the disconnection state. So, those are you don't, for example, in the dream state, we think you have more brain activity in posterior cingulate and anterior cingulate. So if you mix that with unconsciousness, that brain activity is going to decrease, and potentially you'll say disconnection has more of a role from those centers. And that's just what happens when you have to mix up, states to be able to analyze the data, because these are really hard data to get. When Katja told me that they'd done a PET experiment, I couldn't believe it, because it's just unbelievable to me that you could be able to do that. It's so hard to be able to do that. It's hard enough with EEG. To then put them in a PET scanner is phenomenal.
It's actually not difficult with people who are getting the anesthetics, but to get them to fall asleep naturally in the PET scanner was a bit of it involved a lot of praying to the sleep gods and a 30-hour sleep deprivation of the participants prior to the scanning session. So I really liked when you said that you're an impatient person, that you can't wait for people to fall asleep, so you use anesthesia, and I've used to doing it the other way around, and I realized how much easier anesthesia sometimes is compared to natural physiological sleep that doesn't, come when you pray for it.
I was just reading Angus's question there, but it's really about expectation, and that's kind of what you've done, Katja, I think in your experiments, that you have emotional stimuli. That's something that somebody is, expecting in a way and knows about in their world. That if you have an expectation, you might be more likely to encode something under anesthesia. In a way, that's the experiment that you did, right?
Yeah, and in a sense, people who are expecting maybe something bad to happen might be more likely to dream about it and remember, because we tend to remember unpleasant experiences better than pleasant or everyday experiences. Might be able to recall that kind of a dream better. So, in fact, I think big part of anesthesia is also managing the patient's expectations. And what I also think is important is to ask the patients afterwards if they experienced anything, and then explain that it's actually quite normal to have dreams about the anesthesia or the hospital or the doctors during the recovery period. And it doesn't mean that the patient has been awake, because unfortunately, our patients do not know the concepts between connected and disconnected consciousness. They can't definitely tell the difference between having been awake during surgery and dreaming about the setting during the recovery phase. So the expectations of the patient are good to address, and also maybe the post-anesthesia recall of anything, just to put the patients at ease.
I think we'll slowly start winding down, but I noticed a question from Daisuke. Maybe you'd like to articulate it by voice?
Yes. Thanks for the great talk. I'm just curious to know how these two speakers view the performance of the perturbational complexity in detecting disconnected consciousness, because the Sarasso paper appears to show high PCI in ketamine anesthesia. Thanks.
So, I don't have any problems with the paper as reported. The statements, for example, that ketamine produces a dissociated anesthetic state is entirely consistent with, anesthetic literature and anesthetic thinking. And the idea that it's conscious is also consistent with that. The problem is that I've been unable to verify that assertion, because when the patients are at that deep level of ketamine, they just don't interact, and they certainly can't verbalize. And at the lighter levels, when they will interact and verbalize, if they're not vomiting after that high dose of ketamine, which unfortunately happens, is that they're then at a drug concentration which blocks NMDA receptors, and they report vivid experiences, and the same sort of vivid experiences they report before getting to that deep level of ketamine. So I don't have any evidence to say that it's not a dissociated anesthetic state which was associated with dreams. I just don't have any evidence for it either.
I also think that these kinds of findings still need to be verified with the first-person reports. So, for example, in some of the older papers, non-REM sleep was always considered to be a completely non-conscious state, and REM sleep a conscious state. So dreaming always occurs in REM sleep, and it never occurs in non-REM sleep. And PCI between these two states is different, but in fact, the conscious experiences are not necessarily that different. So I think we need to be always careful when we investigate the neural correlates of consciousness, that we are not only investigating a certain physiological state and comparing it to a completely different physiological state without checking whether the person remembers having had experiences. So, and I think this is an we are living in interesting times, both in dream research, but especially in anesthesiology, is that this is becoming more and more important to understand that anesthesia it's not necessarily an unconscious state. It's fine if it's a disconnected state. It's not fine if it's a connected state, but we really are making a lot of progress nowadays in trying to distinguish these two states compared to what the situation was 10, 15, 20 years ago.
Thank you so much.
Excellent. Well, I think we've done about an hour and a half of Zoom.
Yeah. Well, I can I just ask a question, or maybe just a little bit of comment. Also, I'm quite intrigued by Katja's statement about motivation. That's quite unexpected, or probably something that nobody would consider as a critical factor for getting a dream reports, unless you work with these patients or do this experiment, right? And that kind of knowledge, I was thinking maybe able to feed back to some other potentially animal research or application in anesthesia, where the areas that are related to the motivation may be also manipulated to see how it affects on dream or sleep. Like in the case of rats, this could be completely off idea, but we know that rats, when they are stimulated in tegmental area or somewhere, they get conditioned to press the lever until they die and so on. So something like that could be potentially isolated and combined with different kind of drug manipulation or electrical stimulation in the case of the animals to in a sense decompose or reveal some aspects of the sleep or dream, if the motivation aspect is very important. I mean, I've never thought about the motivation and the dream at the same time, actually.
Well, having done a lot of serial awakenings in a sleep lab during the night, I've noticed that if you wake people up constantly, they tend to become sleep deprived, they tend to become irritated, they fall asleep faster, then you wake them up again and again and again and again. And at some point, they might be tempted to say, I can't remember anything, just in order to be able to get back to sleep. So it's I think motivation is an important aspect of dream research, because they know that if they say, yes, I had a dream, they have to describe it, and then they have to respond to 15 different questions about the content instead of just being allowed to continue sleeping. And I would guess that in sedation, something very similar might happen. So if you just don't respond, if you just, you hear it, but you decide not to respond, they will just let you be. And it's quite comfortable to be really drowsy and falling asleep and
but
not wanting to be disturbed by any researcher. So I think motivation is important, and it's difficult to account for because we don't have a measure for it. We can always ask afterwards, but by that time, especially with anesthesia, the participants had already forgotten that they heard, but they didn't want to respond. Possibly.
But that's very interesting from because I'm mainly working on the perceptual side of the consciousness, right? And similar homologous kind of question arises even with the very simple psychophysics experiment. For a long time we know that if we change the reward structure or motivational structure, the resultant sort of threshold or sensitivity actually appears to be different. But in reality, what we are talking is just about sort of how to motivate or how to reward people to say yes or no, and that kind of manipulation, resulted in the discovery or sort of the operational characterization of the non-conscious conscious perception. So something like that could also be incorporated in this type of study. It's gonna be very difficult, but I think it's possible direction.
Yeah, if consciousness was easy, we would have resolved it already.
Yeah.
All right. I mean, I think just to summarize, to conclude from my perspective, I think part of the issue, if you want to try to study this in animals, is also just is understanding the function of sleep, right? There's a function to active sleep, to dreams. There's a function to deep sleep that is evolutionarily conserved. And in a way, we're conflating things by using words that we don't really understand. And maybe by understanding the functions, we might get a bit closer to seeing if there's a commonality with whatever is achieved under anesthesia, where, as far as I can tell, in the data isn't really there showing that general anesthesia achieves sleep functions, right? Neither REM sleep functions nor deep sleep functions, right? So maybe that's another way of studying the problem, is understanding what the functions are for sleep first, and then seeing if there's any drugs or anesthetic manipulations that somehow mimic those functions, rather than thinking about the specific brain patterns that are correlated to those functions. That's another way.
I think this might also be clinically significant in a sense that I recently heard that in the treatment of insomnia, what's now being developed are these alpha-2 receptor antagonist drugs. So, which would basically act a bit similar to dexmedetomidine to induce sleep. And what if we already know that sleeping agents do not usually produce the kind of sleep that fulfills all its functions. So this is also, I think, important in the sense of drug development for treating insomnia.
Yeah. Well, thank you for doing your talks. I mean, I'm glad that you're here in Australia with us, Rob Sanders. And I wish you were, Katja.
Yeah, but we always have Zoom.
We always have Zoom.
Yeah.
Yeah.
Great. Thank you.
Yeah, that was really interesting. I appreciate
Thank you very much for the invitation, and I personally, it was highly interesting for me as well. It was nice to meet all of you.
Thanks a lot.
Okay,
Yeah,
thank you.
Okay, then I'll now stop YouTube. Bye, YouTube listeners. Okay, so now YouTube is finished.